Okay, let's go. Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the 3 NIH-funded National Cryo-EM Microscopy Centers. My name is Alina. I'm an outreach manager and a project coordinator at the National Center for Cryo-EM Access and Training, NCCAT. It's in New York City. I'm joined today by staff members from each center, including my colleague from New York, Joshua. We have Omar from Pacific Northwest Cryo-EM Center, PNCC, and Nathan from Stanford SLAC Cryo-EM Center, S2C2. Before we give the floor to our speaker today, we will give a quick update from each of the centers on the resources that we have available at no cost to the research community. Today's speaker, Puja Majumder, is joining us from the Memorial Sloan Kettering Cancer Center in New York, but Joshua, my colleague, will introduce her properly after our updates. This webinar is a regular event. We host every last Thursday of each month. We highlight users of the National Cryo-EM Centers who share their tips and tricks that have enabled them to use Cryo-EM in their research. You can see all past webinars and the ones upcoming, like we have one already for next month. They can all be found on our shared webpage, cryoemcenters.org. And for final logistics today, if you have any questions on how to access National Centers resources, you can reach out to any of the panelists here using the chat. If you have questions to our speaker, please post them in the Q&A box during the talk. You can post your questions at any point, and we will address them at the end of the talk. Joshua will take care of those. And let's do the updates.
Thanks, Alina. Give this slide just a second to update here. I am Omar Davulko. I'm one of the microscopists at the Pacific Northwest Cryo-EM Center in beautiful Portland, Oregon. At PNCC, we offer our users a seamless workflow from sample prep all the way to 3D reconstruction, including our screening access mechanism where users are invited to come out to Portland and work with our staff for a week to go directly from sample in the tube all the way to structure. At PNCC, we've got a number of microscopes with K3s, Falcon 4Is, all of which are energy filtered, and a suite of sample preparation devices. We also provide training on every piece of equipment that we have, so I encourage everyone to visit us at our website, pncc.labworks.org, to learn how to get access. With that, I will hand it to Nathan.
Hi, everyone. I'm Nathan from S2C2. Just like the other centers, we have the suite of equipment and technical resources from Krios to Glacius and a Tundra on the way. It's currently being installed. We're excited for that. You can grab a screenshot here and learn about our programs. But the thing I want to highlight for everyone is that we are currently planning a workshop at the end of October focused on structure validation. So how do you know you have the right structure that really fits into my map and get the best that you can do? So we can find more information about that on our website. Thanks.
Okay, and I'll update NCCAT. So at NCCAT, we offer data collection, sample preparation and screening, as well as 2 types of training. So very, very similar to other national centers. We have quarterly deadlines for proposals. In fact, next one is coming on October 1st. All of our proposals go through the external review committee before we can allocate any services, and better scored proposals will have a priority when it comes to scheduling. All proposals submitted to us have a 2-year lifetime after the submission. And just a quick update, in terms of our workflow, we have incorporated the use of coded grid boxes. So in case you are an active user here at NCCAT and you have not received the coded grid boxes, please do reach out to us and let us know. Thank you. That's it for the updates. Now I will ask Joshua to introduce today's speaker.
Hello, everybody. So today we have the great pleasure to hear Dr. Puja Majumder, and she is currently a postdoctoral research associate in the Department of Structural Biology at the Memorial Sloan Kettering Cancer Center here in New York in the lab of Patel Lab. So to give her a little bit of background who she is, she completed her PhD at the Indian Institute of Science in Bangalore and officially graduated in June 2021. She joined MSKCC on April 22, and she became a user at the New York Structural Biology, where NCCAT is hosted on June of 2022 as well. So it's a great person to have here, and we have come to know each other very well. Her talk today, is titled "The Structural Basis of Filamentous CRISPR Effector Revealed by Cryo-EM." And recently, she has been selected to present a talk on the New York area Cryo-EM meeting on June of 2025, which I heard it was a great talk. And also, she was selected to give the keynote speaker at the Electron Microscopy Society of India. So clearly, a lot of people are interested to hear her research as we are. I asked her a fun question about which one is her most recent book that she read, and she said that her most recent book was Before the Coffee Gets Cold by Toshikazu Kawaguchi, but in reality is, more science books of enzymes chapters of the biochemistry textbook. So if you want to see more of her research, please, you can find her GitHub by putting her first name and last name .github.io, and you will be able to see a lot of her papers. So with that, Puja, I think you can take it away.
Thank you, Joshua, for this kind introduction. I'll now share my screen. Please let me know if the screen is visible. Okay. I'll start today's presentation by acknowledging the people who have contributed and supported throughout this work. First of all, my postdoc supervisor, Dinshaw Patel, and my collaborator, Dr. Luciano Marraffini, and an extremely talented graduate student, Christian, who has contributed to the microbiology part of this work. With this, and also I would like to thank the Cryo-EM facilities, MSKCC, NCCAT, and NYSPC, where these data sets were collected. With this, I start today's talk on filamentous CRISPR effector. CRISPR systems are antiviral defense systems. Viruses are common pathogens to all forms of life. We humans have our own virus, plants have their own, and bacteria is no exception of that. And they have their viruses, which are called bacteriophages. And interestingly, like us, bacteria has also developed a strong, diverse immune system against these viruses. And the immune system associated genes of bacteria, they tend to cluster together, forming defense island in the bacterial genome. Now, in this defense island, people have found that there are innate immune systems, also adaptive immune systems in bacteria. There are more than 150 new innate immune systems that have been discovered very recently. And interestingly, they are named as a god and goddesses from different mythology, like Lamassu, Azaka, Zoria, and so on. But there is only one adaptive defense system that we know so far is CRISPR system. And as you can imagine here, bacteria is using different kind of strategies to fight viruses. But this diverse range of strategies can again be divided into majorly two modes of immunity. One is individual immunity, in which case the host bacteria, the infected bacteria, survives. This can happen by direct attack of the viral nucleic acid. And another mode of immunity is population immunity. In this case, the infected host bacteria is sacrificed, but that also prevents the spread of the virus to the population of the bacteria, and rest of the population survives. This mode of immunity is also termed as abortive infection mechanism, and the defense system that I'll be discussing today follows this population immunity or abortive infection mechanism. Now coming to CRISPR system, like other CRISPR systems, type 3 CRISPR also follows two steps of immunity. First, immunization of the bacteria, and second is the immunity provided by the bacteria. Now at the immunization step, small fragments of the viral DNA gets incorporated into the host genome, forming CRISPR array. And when next time the virus infection happens, the spacer sequences from this CRISPR array gets matured to CRISPR RNA or guide RNA and guides the Cas nuclease complexes to recognize the viral nucleic acid. Among these Cas nucleases, Cas10 has a very unique role. It can act as nuclease, also as a cyclase. So the cyclase domain of Cas10 then synthesizes cyclic oligoadenylates. These small cyclic RNA molecules act as a signal to auxiliary effector proteins, which we are interested in. These auxiliary effectors have very simple bipartite architecture. They have a sensor domain, which has a CARF, which are known as CARF domain, CRISPR-associated Rossmann Fold. This sensor domain can sense this cyclic oligoadenylate signal molecule, these RNA molecules, and give the signal to the effector domain, which are majorly nucleases. The nucleases then cleaves the nucleic acids and causes the growth arrest of the infected bacteria, and thus prevent the spread of the virus to the rest of the population. Interestingly, what we have found that there are several CARF containing effectors which do not have any nucleases associated to it, rather have enzymes like adenosine deaminase, different domains like TIR domain, or different transmembrane helices associated to it. So we were very interested in these effectors, and today I'll be discussing the work with CAT1, CRISPR-associated TIR1 effector. The question we asked is that if this CAT1 system has any antiphage activity, and if yes, how it provides the antiphage activity. Now the first question was answered by Christian when he shows that CAT1 and 10 of its homologs provides antiphage defense against 6 different phages. And then we started our structural analysis with this protein. We purified the protein, and with a putative substrate of the TIR domain NAD, we incubated the purified protein and shown that by Sekmal's analysis that this protein forms a dimer in solution. Next, we wanted to screen which cyclic RNA molecule can be the activator of this system, and we screened a whole library of cyclic RNA, and we found that cyclic tetra-adenylate can bind to this protein very tightly. And then we verified that indeed cyclic tetra-adenylate is the activator of this system, because in presence of cyclic tetra-adenylate, CAT1 can convert NAD to ADPR and NAM. Now we started our sample preparation and grid freezing for this active state of CAT1 defense system. I must tell you that this was the rate limiting step of this project, the sample preparation and grid freezing. As we started with, when we incubated the small RNA cyclic RNA molecule with the purified protein and incubated for 1 hour, we see something like this. This is the low mag image of the atlas, where you can see the squares, and you can see the sample was so thick, it was forming this black patches on the square, and we could not even see the holes. Then we started optimizing the sample preparation conditions, and as we hit this condition, when we added CA4, the cyclic RNA molecule, and within 10 seconds we froze the grid, and on the grid we started seeing this kind of filament assemblies. And during grid freezing, we also used a positive blot force using FAI Vitrobot, which helped us to get these kind of filament assemblies on the grid. Also, this is a representative micrograph, and some of these micrographs have this kind of spread of filaments, but some of the micrographs were empty, and some had very less amount of filaments on it. So we had very heterogeneous particle distribution throughout the dataset. Hence, we collected a larger dataset for this project. We collected around 10,000 micrographs. Next, I'll show you how I have processed this filament dataset, starting from particle picking, selection of box size, and how I cleaned up the particles, and then 3D classification and refinement job. Now, this here you can see a micrograph where most of the parts are empty, but we only see the filament at the bottom of the micrograph. So I decided to start particle picking by manually, and I manually picked around 2,000 particles. I did 2D classification, and here you can see the 2D classification was encouraging because at the side view you can see the repeat units, and these are the front view of the filament. Next, I used these 2,000 particles to train the model for particle picking using Topaz. This is the output of Topaz training, where it shows you as you increase the number of epochs, it trains the model and increases the picking average precision, and it reaches to plateau when the epoch number was 8 to 9. That means the model was well trained. So I used this model for particle picking, and as you can see, the Topaz particle picking did a very good job, and it could pick around 3 million particles from 10,000 micrographs. The one problem I faced with Topaz particle picking is that it picked up a lot of duplicate particles, but I could remove these duplicate particles using duplicate particle remove job in CryoSpark. And I also used a filament tracer job for this project. That job did not work well. Topaz worked well for this project. Then with these particles, I used different extraction box size because at this point we did not know what is the size of this filament. So I started with 256 pixel box size, and the particles were not aligned, the filaments were not aligned properly. As I used 512, I could see the filaments were aligned properly. As you can see in the side views, there are repeat units, and the front views are also present. And I also tried to use the larger box size than 512, but I did not get additional any information, so I stick to 512 box size for this filaments. Then I did 2D classification job, and here we could see the side views and the front views, but at this stage I did not discard any particle from the 2D classification job. Rather, I used particle cleanup using hetero refinement job. This trick I learned from Dr. Richard Hyde from MSK. This process has 3 steps. Step 1 is building an ab initio model using the good 2D classes that we have, and I try to maintain similar particle ratio for different views. And in the step 2, I used another ab initio model building job, and then create noise 3D volumes, at least 4 to 6 classes. What I mean by this, I will be describing that in the next slide. Now coming to step 3, here I use the volume from step 1 and 2 as my input volume, and put all the particles, total set of particles that I want to clean up in the hetero refinement job. Now coming to the example. So what I have done here is, this is the first round of ab initio model, and here you can see the repeat units and the pentagonal filament assembly at the middle, and here is the trigonal filament assembly. Next ab initio job was done, which created these noise volumes here. The way I create these noise volumes is that I start the ab initio model job and kill it very quickly as soon as each volume has more than 0 particle number. And after killing the job, I mark it as complete and use this volume as the input for the next round of heterorefinement job. And I use the total set of particles which we would like to clean. The output I got from the heterorefinement job looks like this. The ab initio model improved significantly. Here I see the repeat units, the pentagonal assembly of the filament network, and here is the trigonal assembly. And it helps us to clean up all the noisy particles with the help of these noise volume classes. And this step can be done iteratively as long as we want to clean up our particle set. Once we are happy with the cleaned volume map, I performed the 3D classification job. In the 3D classification job, class 1 and class 2 looked well. And I used class 2 and for the further refinement job and used non-uniform refinement, and it refined the map at 3.4 angstrom resolution. Now this is my total map, the total volume, and for the molecule, for the building, I use this cross section which is colored in purple here. And using that, here I show the protein parts of the filament where I can see very nicely the side chain densities. And this is the density for the cyclic RNA signal molecule, cyclic tetra-adenylate, and the density was good for all the parts. Now I will go through the filament structure that we found from this map in a step-by-step fashion. So what we observed is that Cat1 dimer stack on top of each other, forming this filament assembly, and the filament is glued by CA4 signal molecule at the middle, shown in yellow. And the CARF dimeric CARF domain forms the center of the filament, which is the sensor domain for this defense system, and the TIR domain, which is the effector, are radially arranged. Now, if we take the single dimeric unit and rotate it by 90 degrees, we see that each Cat1 monomer forms present in a twofold symmetry. And if we rotate the whole filament assembly, we see that there is a twist. The next observation was in the 2D classes, we see in the front view that these filaments can form trigonal filament assembly, 3 of the filaments coming together forming a trigonal assembly. And these are the side views where one can appreciate the repeat units. As we solve the structure of this trigonal filament assembly, what we observe that the TIR domain of filament 1 interacts with the CARF domain of filament 2 here. Even though filament 1 interacts with filament 2 and 2 interacts with 3, filament 3 was not interacting with filament 1. Rather, there is a tilt along the central axis in filament 3 that brings the blue TIR domain below the plane than filament 1 and 2. And that gives rise to a spiral helical bundle with a trigonal pore in the middle, and the filament could extend in both the sides as shown by this green double-sided arrow. Now I would like to draw your attention to this black mark, black triangle here. Here you can see that the CARF domain of filament 3 is now free. That means the TIR domain from the adjacent filament can interact with the CARF domain here and have an opportunity to form even larger filament network. And that's what we see next. We see that 5 of these trigonal filament assemblies are coming together and forming this pentagonal filament network. This is the front view, and here you can see the side views. As we solve the structure, what we see is the tilt along the central axis of filament 3 now brings this magenta TIR domain above the plane than rest of the filament, and that gives rise to a pentagonal spiral bundle with and that spiral bundle can be extended in both directions again along this green double-sided arrow. So then we asked what is the biological significance of this huge filament network formation. in order to do that, we decided to break the interfilament association by mutating the interfilament as interacting residues. As this was backbone interaction, we deleted one residue in one case from filament 1 from the CARF domain and 2 residues from filament 2 from the TIR domain. And we checked that by mutating these residues, we could actually prevent the network formation on the grid. And this is the zoomed view. While we can see that there are small filament fragments present on the grid, but the filament network was completely absent. Then we performed our functional studies with this interfilament association breaking mutants, and we find that more than 80 to 90% activity of this system was abolished, suggesting this filament network enhances the activity of this defense system. Further, we wanted to understand the reason of multimerization of Cat1 dimers. In order to understand that, we solved 2 more structures, one with non-hydrolyzable NAD analog, BAD, and another one with NAD itself. What we found that both BAD and ADPR, which is a product of NAD hydrolysis, in both cases, these molecules are present in between 2 TIR domains. As we zoomed in, we see that there are several residues from the top TIR domain forming the present as the NADase pocket, and there are 2 residues from the bottom TIR domain which forms the NADase pocket, suggesting that the NADase pocket is a composite pocket, requires the dimerization of the Cat1 dimer, like the stacking of Cat1 dimer. And we verified that each of these residues are important for the activity of this protein by single site mutants. So far, summarizing the talk, what I have shown you is that once the phage infection happens, the CRISPR system gets activated. Cas10 produces cyclic oligoadenylates from ATP. Among this cyclic oligoadenylate, cyclic tetra-adenylate is recognized by Cat1 dimer. And once Cat1 dimer recognizes its signal molecule, it forms this huge filament networks formed by pentagonal and trigonal filament assemblies. And if we take a single filament and rotate it by 90 degrees, we can see that the Cat1 dimers are stacked on top of each other, glued by cyclic tetra-adenylate in the middle. This forms a composite NADase pocket in between the TIR domains. Once the composite NADase pocket is formed, the NAD comes and binds to this pocket and gets hydrolyzed, and that causes NAD depletion and ultimately bacterial growth arrest because of depletion of an essential metabolite, and that prevents the viral spread. With this, I thank you all, and I'm open to any question.
And I think if people have questions, we can make them available to talk. So as the hands come up, I will allow them to talk. So go ahead, Abhinav.
Hi, Puja. Can you hear me?
Hi. Yes.
Hi, great to hear from you after so many years. So, fantastic. So I had a couple of questions. So first is regarding the timescale of these events. How does that work? Have you tried doing, sample freezing at different time points with the RNA and see how the typical timescale of this assembly is?
So the time scale, when we increase the time scale more than 10 seconds, this is what happens. It forms very thick sample.
Okay.
But the exact time frame, how it progresses to form this kind of network, for that we are trying cryo-ET. to understand in vivo how this filament assembly is forming and also the disassembly, but we are still trying that. But at this point, we just know that it forms within seconds.
And presumably the reason why you think a filament formation is required is to essentially have multivalency and essentially do the NAD depletion more efficiently, you think?
Yes, yes. That's what we think.
But what I was trying to understand is why it would need to form all these very specific ordered structures for that same purpose.
Yeah, that's not very clear, because when it forms a single filament, it still have here. Even the single filament still have the active NADH site. Why it has to form interfilament association and those specific structure is not very clear to us. What we have seen that once it forms the filament network, it stabilizes the filament and hence the composite NADH pocket. But beyond that, we don't know yet.
Okay, thank you.
Thank you.
All right, you may continue now. Tina?
Yep. Hi, great talk. You mentioned a little bit in your response to the last question about the disassembly. How do you think that happens? Like, you know, presumably the cell clears the infection. Is there a way for this to disassemble? Like, how stable are these filaments?
So how the disassembly happens, we don't know yet. We are trying in vivo cryo-ET, but we know that it disassembles because we have seen in vivo data. We have in vivo data that NAD level. So these are growth arrest. These are not killing the cells. So once it clears the infection, the bacteria starts regrowing and the NAD level shoots up. That's why we know this filament network disassemble after clearing up the viruses. But how it actually happens, we still don't know.
Got it. All right. Thank you.
Thank you.
Does the panelists? Oh, I see. Okay, so we also have a couple questions in the Q&A chat box. Let me go with the first one from Michael. The question is, Puja, let's see, you're ready. Yeah. How long are the filaments on average, meaning how many oligomers?
How many oligomers?
Yeah, or how long can these filaments go?
So theoretically, I think it can form a very long filament. As you can see here, there are several repeat units. But what I have observed is that after certain range, that filament starts breaking up. So with that, what I see here is kind of 1, 2, 3, 4, 5, 6, 7, 8, around 10 repeat units that we can see in our total map.
And do you now, as a follow-up, do you expect that to be different in the cryo-ET in the future, or this is it?
So we are interested in cryo-ET to understand how these filaments assemble. Like we are seeing single filament, and then these single filaments are interconnecting with each other, forming a network kind of a spread. So we were interested to know how it forms this filament network, and also how it dissociates. Because that is very important, biologically relevant to dissociate. Otherwise, bacteria can't survive again after the infection is over. So these are the questions we are asking in cryo-ET experiments.
Okay, so good. All right, so we have more questions coming in. Also, this is the next one. Could you comment on any preferential orientation that you observe? If so, seems filaments have this distinct directional preference but your 3D models are looking pretty good. So do you have any preferred orientation?
So yeah, actually, what I haven't mentioned that here, if I tried to discard particles or select particles in the based on the 2D classes, I see preferred orientation. But when I use this method for particle cleaning, the hetero refinement method, my preferred orientation problem was solved. So that actually helped me a lot with getting all the different views. So with that, I could get kind of good 3D map.
And there was another question. What are the resolution of those NAD and BAD bound structures? Are they also 3.4 or?
No, the BAD bound structure, I think it was 3.6 angstrom, and the NAD bound structure was 4 angstrom.
Great talk. Just out of curiosity, was the filament formation expected before starting cryo-EM on this protein? I was wondering because the protein itself is quite small.
Yes, so the protein is actually 29 kilodalton, the monomer. But we were very interested because the TIR domain, which is also present in our innate immune system, also plant innate immune system as well. So in this human or in plant immune systems, TIR domains tend to form this kind of filament assembly. Very different, but tend to form some kind of filaments. So that's where we started, and we find this in bacteria.
I have a question of my own. Why do you think that topaz work better than filament picking?
Yeah, actually, filament tracer did not even pick the micrographs where the filaments were very densely populated. I don't know why exactly that happened, but the topaz worked really well. The reason I don't know, but filament tracer did not work.
I think we have a couple of follow-up questions. Let me allow them to speak because they raised their hands. But you can also put on the Q&A. I'll do Abhinav first. Go ahead.
Just add to the discussion that you are having. Often what I've seen is when you have a twist like this, like a turn on the filaments, because I've processed filament data with CRISPR, and I've seen when they have a slight turn like this, they find it very difficult to align. One way to do that is to do, there is a tau filtering, or there's some filtering that you can do, which will specifically disregard filaments when they tend to have this twist, like a C. You see, most often you see in your 2D classes.
Oh, yeah. I think, yeah.
So yeah, that's why probably topaz works, because it comes in with no bias.
Thanks. Yeah.
Thank you. Yeah. I didn't know that.
Yeah.
And there was another person have the hands up, but maybe they raise again. A question came in is, how could you differentiate between NAD and ADPR at that resolution?
Oh, so ADPR does not have one. This, if you can see here, so the one moiety is absent in case of ADPR, so the density is smaller. So I could not fit the full NAD molecule in that, but I could only fit the ADPR molecule in that density that I had. The density is not very good, and for TIR domains, previous previous manuscripts or published data also says that as this composite pocket is not very stable, so you don't get very good density for the NAD moiety in there. So that's what we got, and that's how we modeled.
All right. I guess I will ask another question that I have. This protocol of heterogeneity refinement that you did.
Yeah.
I'm actually very curious because it's different from how I normally do it. Do you think it just works good for filaments, or do you think it will work well also with other techniques, other forms?
I use this method for currently all my data sets, and it works very nicely actually, because it helps to separate all kind of noise, because we are giving these volumes as input, the noise volumes. It helps to separate all kind of noise from the particle set. So it works very well for all kind of data sets. For membrane protein data set, I have used this method. It works very well. For soluble protein as well, it works very well.
I will definitely give it a try with my own data sets.
Yeah.
Are there any other questions from the attendees? If not, well, I don't know if there's any closing remarks from the centers, but thank you very much for giving the talk, Puja. Very interesting.
Thank you.
Thank you.
Thank you, Puja. Well, I think this brings us to an end. Thank you, everyone, for joining us today. And please reach out with any questions. We're here to support your science and help you with your cryo-EM journey.